Our next topic is going to be how we generate the diversity that we have associated with our antibodies, and kind of in part the diversity that we end up with our B cells, because it's our B cells that are going to be making these antibodies, all right? And that process of generating diversity in your immune system is because of the process of VDJ recombination, all right? It almost sounds like it should be something perverse, but it's not. In terms of VDJ recombination, all right, essentially what we're talking about is we have a whole bunch of genes that are responsible for making those antibodies, okay? And in terms of VDJ recombination, this was a topic that drove me absolutely nuts as an undergraduate because it felt like the instructor spent days and days lecturing on VDJ recombination. And what I eventually put together on this that would have made it a lot simpler for me to understand is essentially what's going on here. Is we've got some alternative splicing that's going to occur with generating our antibodies, okay? So remember with our antibodies, I'm going to draw it over here, all right? I drew this up. We had the heavy chain that I'm drawing in pink, the light chain that I'm drawing in blue, all right? Essentially what we've got going on there, all right, is we have a series of genes, V1 through, I forget what the number is. I think they're like 12 or 15 of them. It doesn't matter, essentially. But you've got genes that are responsible in there. You've got genes that are now going to go D1 to, I think that's another one that's high, 15, 12 maybe. We'll make it less. And then we've got J. And J, there are fewer of these. This I remember. Okay. And then, what color haven't I used? Blue. All right. We've got, I've got to have another shape here. Let's make them this way. Rectangles. We've got C, all right? And in terms of that, these are going to be the genes associated with our, let's go with pink, with our heavy chain, okay? Then in terms of that with our light chain, we're going to have a similar organization of genes, all right? We're going to have V1, and I forget the number. Oh, these go into the hundreds. Okay? We've got D is next, all right? And that's going to be a difference between our light chain and our heavy chain. There's no D, all right? We still have J. We have fewer options with J. I guess I can just draw them all in. There's J1, J2, J3. All right? And then we've got one option for a constant region, okay? So in terms of this, these boxes that I've drawn here represent genes that are associated with the variable region. Goodness, I can spell. I promise I can spell most days, just not today. The variable region, okay? And in terms of what you've got going on here, there's a lot more variability in the light chain than there is in the heavy chain, okay? You've got a missing portion associated with the heavy, or you've got a portion associated with the heavy chain that's not found in the light chain. That's the D region. Both are going to have a J region. Both are going to have a C region. So essentially what happens and what you've got going on is you might have to make up your heavy chain up there. Say this ends up being variable 6 region. All right. The D region is unique to the heavy chain because that's what gives rise to our hinge region, okay? And I'm going to kind of draw it like this since it represents a hinge, all right? And we'll say this is D4. Then we're going to put a J, J1, and then the rest of our constant region. All right? So essentially what this does is this makes one heavy chain. All right? And in terms of that, essentially what you're doing is you're splicing together one gene from the variable region, one gene from the D region, one gene from the J region, one gene from the C region. All right? And that is what makes up your heavy chain protein. Now in terms of this, I just picked one random combination. But this is how we get the variability in the thousands of different combinations of antibodies that your body is capable of making. Because this variable region now, if we change this, this could be V2, D4, J1, C2. It's going to recognize a completely different antigen than the one I have drawn up here. Okay? And in terms of our light chain, we get the same thing. All right? Our light chain, we're going to have a variable region. We can call this V1. J3, C1. All right? And essentially how this is going to come together to make this little antibody that I've made up here. So we've got pink, green, and blue. Okay. This is now corresponding to this region here. All right. Then we've got our heavy chain that we made over here. We've got our variable region. We've got our hinge. We've got our J region. And we've got our constant region. So in terms of that we have here are going to correspond to these two chains. All right? And in terms of our antibody, what makes it unique, all right, is that variable region is going to be the portion that will bind to our particular antigen. So if we take this out and make this a triangle, all right, then we can, with our light chain, maybe this has got a slightly different shape, all right. What that's going to do this is going to be where our antigen is going to bind. So in terms of our epitope or our antigen, this would be the combination that we would need to have this particular antibody recognize that particular antigen. All right? And if we change out the gene, we get a different heavy chain, different light chain combination. All right? So essentially, within the B cells in your bone marrow, you have all of these different splicing options happening. All right? And it's the splicing options here with VDJ that result in the diversity of heavy and light chains that give us all the different types of B cells that we have. All right? So in terms of that VDJ recombination, this is crucial in generating the diversity in your immune system. All right? And what genes you have here in the variable region, the V region, the D region, the J region, the C region, that's what you inherit from your mom and dad. Okay? There are a ton of different variations in the genes that you can inherit from your mom versus your dad. So in terms of the health of the offspring, the more differences you have in these genes associated with this process, the more different combinations you can make. And in terms of having more combinations that you can make, that's what's going to give you a stronger immune system, because you're going to be able to recognize more antigens and being able to fight off more potential pathogens. All right? So I know VDJ recombination is a little bit abstract as far as understanding it. What I want you to take away, essentially you've got some alternative splicing going on. All right? So essentially what you're having happen is you're going to take one V gene, one D gene, one J gene, one C gene, and you're going to splice those together to make one messenger RNA that is then translated to the protein that makes up part of your antibody. In this case, on this side over here, this would make up the protein associated with the heavy chain. If I did that splicing over here, say we do V1 to J1 to C, that would give you your light chain. All right? Hopefully that makes sense. If it doesn't, please ask me questions. Otherwise, I hope you have a